Hello everyone, I'm Nora Castner from the Human-Computer Interaction Group at the University of Tübingen, and I will talk to you about MinHash, a fast approach for scanpath classification. Scanpath approaches that consider the whole scanpath often suffer when it comes to real-world tasks. For many tasks such as driving, performing surgery, or even grocery shopping, there can be numerous areas of interest. This becomes more problematic with the longer the time on task, which contributes to the larger data stream, for instance, the sequence of AOI transitions. We present a method, MinHash, that uses the same name technique to extract scanpath similarity from subsequence frequencies. This method improves on the runtime performance compared to traditional sequence alignment approaches and is highly generalizable due to the models built using data mining. We have also made the algorithm publicly available on GitLab. First, we construct areas of interest, or AOIs, and this can be done as the researcher pleases. We then encode the fixation sequence based on the AOI hits. This gives us a normalization of the scan path to spatial and/or semantic information, but maintains the temporal aspect of the scan path. And though we did not test it, you could also include fixation duration with, for instance, temporal binning. We continue on to get the sequence for the whole scan path. And up to this point, our approach does not differ from many other state-of-the-art sequence alignment approaches. The next step is to extract and compare subsequences of scan paths. For this, we use n-gram scanning to extract words of a fixed length using a sliding window over the scan path. For instance, we can look at words that are 4 letters long. We deliberately insert gaps to compensate for signal noise. This way we can map similar patterns to each other, even if there's a slight offset in the signal. The words then get tallied into a dictionary. And matches affect the frequency of the words in the dictionary. Now we have a dictionary for each scan path. The traditional approach is to consider the dictionary as a high-dimensional vector, and generally, for comparing two scan path dictionaries, you would calculate the similarity, for example, by the Euclidean distance. However, as previously mentioned, large and AOI complex scan paths can create excessively large dictionaries. Our solution is to compare the minimal response of a set of hash functions over the dictionary, MinHash. This corresponds to a repeated comparison of randomly selected subsequences of two scan paths, and the likelihood of equality corresponds to the Gjergji index. Now, for all the scan paths, the straightforward MinHash approach is to count the matches between words or hash functions. If you use m distinct hash functions, it takes m operations to compare two scan paths. But how do we handle when there's different frequencies within the scan paths? To solve this, we use local sensitive hashing. For scan paths that have min matches for two substrings, we can visualize the substring frequencies along two axes. We apply some random thresholds, and this creates a signature. And scan paths with similar frequencies will appear more often on the same side of the threshold. The similar frequencies found through the thresholding are then used to estimate the Gjergji index of two scan paths. Then, using the Gjergji index, we can take unseen scan paths and classify them using a k-nearest neighbor classifier. To evaluate MinHash, we use the dataset provided by the 2019 ETRA challenge. Subjects were given 3 tasks: free viewing, puzzle, and Where's Waldo, which provide distinct, task-based scan paths for classification. We evaluated MinHash with 4 different minimum hash functions: 64, 128, 256, and 512. And compared the classification performance to Subsmatch, another state-of-the-art by Kubler and colleagues in 2017. This method also employs subsequence frequencies for classification. From this plot, we can see the accuracy of MinHash and subsmatch. MinHash, in green, is in the 80% accuracy range, with the best performing at 88% accuracy, and that's using 256 hash functions or 512 MinHash functions. And this is comparable to the performance of subsmatch here in red, which is about 91% accuracy. But what stands out from MinHash is the time that it takes to accurately classify the different tasks. As you can see here on the x-axis, MinHash is much quicker than subsmatch at classifying. Subsmatch clocks in at around 29 seconds to classify all the tasks, whereas MinHash with 64 hash functions takes about 5 seconds. But if we want that good accuracy, that 88% accuracy, it'll take us about 15 seconds. Additionally, MinHash has a constant runtime for long scan paths, which is not the case for subsmatch, and is definitely not the case for sequence alignment algorithms such as the Needleman-Wunsch. Thus, we can conclude that we have fast subsequence similarity extraction and classification, because the number of hash functions affect the runtime. And this is applicable for long and complex scan paths, and it's also highly generalizable since it uses data mining techniques. Thank you for your attention.